Okay, hello everyone. This is a pleasure today to introduce our today's speaker, Anna Beckenhaus, who did her PhD at the GIC in the UK, and then she joined Garda in Morocco to work there and develop her group for some years. And she recently joined the IPK Institute in Germany as a group leader for resource genetic and reproduction in the genebank department. In this capability, she oversees the management and regeneration of the IPK ex situ collection of legume, cereal, vegetable, and medicinal plants. So our work today will delve into advancing the use of genomic data in gene banks, and our today's seminar title is Leveraging Genomics Resources in Barley Pre-breeding. So thank you, and we're happy to listen to you, Anna.
Yeah. Hi, everyone. Thanks, Sebastian, for the kind introduction. And it's really nice to give this seminar today in my second week at IPK, where I'm kind of at this crossroad, right, of now reviewing what I've done in the past, but probably also how that's influencing how I think about the future work at IPK. So with no further ado, I will start the presentation. I hope you can see everything nicely. Yes. So as I mentioned, the last 3 years, I spent a lot of time sitting with breeders and the genebank team at ICARDA in Morocco, trying to think how we can maybe optimize and also make pre-breeding more targeted. Because so far there's been a lot of pre-breeding efforts done by teams that were either coming more from the research side or maybe also from the breeding side. But the idea was really to build that connectivity between the guys that know gene banks really well and the guys that know breeding really well. And we kind of came up with this pipeline where we said, well, at the beginning, you really, for pre-breeding, need to sit with your breeders, kind of with your stakeholders, and ask them, well, what are really traits that you need? And the trait identification is maybe the most crucial part to any step downstream, because you can't handle too many traits, and it's still very expensive and time consuming to do pre-breeding. So really you should possibly focus on traits that are A, very much needed by farmers, but B, also the breeders can't deliver on right now. So that would already focus the traits quite a bit. With that knowledge, you can then really go into the collection of the gene bank. And in this case, so you can go to the gene bank characterization. And this pipeline I present here works probably specifically well for land races, maybe less so for crop-wide relatives. We'll come to that point later. And then a lot of what we thought about was really, well, how can you now come from these 30,000 land races of barley that we hold at ICARDA, for example, to a handful that we can actually phenotype. And here you can really leverage new data platforms such as climatic data, sequencing data, but also, of course, phenotypic data, to then get from thousands to hundreds to maybe a handful of land races that you can really cross. And here's kind of where you need to overlap with your breeders again as a pre-breeder, because you really want to cross to the right elite. And in land races, we think that's really the newest elite, right? So you should replenish your pool of kind of the elite variety that you use in your crossing scheme every couple of years, so that you don't have this kind of loss of genetic gain in pre-breeding, where you then always lag behind by maybe 10 years just because you found one line that you really like to cross with. I can understand that. I had the same, but it's really not the most efficient way to go. And then you enter this very well-known spiral of population advancement, where, again, genomics can really play a big role nowadays. But also it comes back to the trait you identified, right? So you really need to build up your testing sites and your protocols. So one example I think here that was also really fantastic to be involved in is to think about global kind of networks or platforms for testing. So I know, for example, in the wheat community, Barbara Steiner is trying to build up a targeted testing site for fusarium, which is something we maybe all been doing ourselves, but doing that as a global team and really finding the best field conditions where we can really test for the trait of interest will be really important. The same for salinity. We really put a lot of effort in building up field testing sites where we can screen for our target trait. and not just for general performance, because we will never beat breeders in that. And yeah, then after about 5 years, if you really optimize it and run it smoothly, you should have a variety that genetically is very similar to your elite that you started with, but has small parts from the landrace that really improve the specific trait. So yeah, as I said, my biggest struggle was with this question of how to get from thousands to a handful. So here we can just see the wide variety of Icada barley land races available, and where they were collected from. And classically, what pre-breeders and also I think researchers have done a lot is to try to make a core collection of maybe a few hundred to thousands. So one example could be here that you use the passport. That was maybe more old school way, right? That you say, okay, we have this really large diversity of barley here. Diversity is largely driven by geographical patterns. So if we take some lines from North Africa, some from Europe, some from Asia, some from the Americas, and maybe some from India, we probably cover most of the diversity there is. Or also we could phenotype and then kind of go down and down and down. But of course, genotyping has really changed here how we make these core collections, where you would now say, okay, we genotype everything, we map it out where the diversity lies, and then we try to make a subset that is as small as possible while capturing as much as possible of the genetic diversity. And there's very nice codes that can do that for you. And that's great, but there's one issue which Icada has identified very early on from that was mostly driven by this work here on the left from McKay from 1995, so a long time ago in Australia. People already thought, well, if we are looking for a specific trait, why are we looking at all of the diversity if some of the lines have absolutely no chance of having any resistance against the trait of interest? And the example they really elegantly showed that this works is with boron toxicity. So they were looking for boron toxicity in land races and realized, well, actually boron is quite high naturally in soil in the Mediterranean region. So possibly we should only take lines from the Mediterranean region, right? It seems so simple and based on these really basic evolutionary Darwinian insights into biology, but it just hadn't been applied to gene banks yet. And yeah, so this scheme or this idea is known as focused identification of germplasm system, or short FIGS. So the idea here is that if you make a FIGS subset, you actually increase the likelihood of finding resistance to the trait of interest. And at Icada, that was also tested with actually in barley. So from the gene bank, they created a random set of lines. So really just rolling the dice, and then added a disease screen and found that 10% of this random set are resistant on average by chance. And then if you make a core collection using all your knowledge about the diversity, you're still at about 10% because you just didn't, increase your chances. But if you do a fixed set, you're actually at 17.8% of the population being resistant. Still doesn't sound so high, but 17.8, right? But it's actually nearly double. And double means half the cost, half the effort, with double the chances of then finding what you're looking for. So in barley, we applied this mostly also for salinity, because it's again a soil trait that's very stable. But going forward, of course, you can use any of the climatic variables that are there. Just the whole system relies on you having really good passport data. It's kind of an underlying issue. But because all of this is about genomics, I'd now like to show also how we try to now combine the advances we've seen in kind of genomics and using this bioclimatic data, which we're used to from FIGS. So here again, you can see the map of the barley genotypes from the Icada collection, but only 1,700 that were genotyped using GBS. And we can see again our nice subpopulations clustering by geographic drive. But this time we said, okay, so if we would want to find specific QTLs now, and actually understand something genetically about the trait, we would have to phenotype all of them, which is a lot of effort and very costly. But what we have available for free are these bioclimatic variables that we use in FIGS, right? So if you go to the World Clim website, you can download a lot of variables for every place on Earth, such as annual temperature, monthly precipitation, and also temperature and precipitation extremes. If you mix them together, you would run a GWAS, or what we would then call an environmental GWAS. And here's just one of the examples. We're still working through this data set because there's a lot of cleaning you need to do on both sides, specifically though the passport data. So if you have, of course, lines that don't actually come from where it says so in their passport, it really messes up with your results. But one example I already wanted to show is where we only used the Hordeum spontaneum, so the wild barley accessions, and looked for annual temperature fluctuation. Again, this is quite a small set of a few hundred lines, but you can see that here each dot is on the map is one of these spontaneums, and you can see that there's kind of a shift between areas with very high temperature fluctuation being, especially of course towards Central Asia, and the Mediterranean population having evolved under much lower fluctuation. And we identified this QTL on chromosome 1H, which is overlapping with actually not that many candidate genes, and one being ELF3, which is really interesting from kind of a flowering time network understanding, because ELF3 is known to be involved in circadian rhythm adaptation, and in other crops has actually been linked to adaptation to high temperature fluctuation or altitude. So this is something I would really like to dig deeper, and also actually develop these environmental indices we can use to be more precise to the crop we work with. Okay, so I've talked a little bit now about what we did here with the environmental data. Now we also move into an age where this was all still run on GPS data, right? So mostly marker data. But then there was this publication of the Watkins collection in wheat. And we said, well, this is really interesting because you actually have whole genome sequencing data available, and you can again do your nice population clusters and so on. But from the fundamental research side, it was also really interesting because you could now look at your haplotypes. So here on the right, we have a map where you can see the haplotypes. So from left to right, we go across the chromosome arm of 2A of wheat. And as a reference, they used SY matis, which is known to have this resistant QTL known as AVR-RMG8, which carries the PM4 highlighted in yellow. And they use this data to fine map, right? So then going down, you have the different accessions of wheat, and you can see actually, oh, wait, some of them, for example, the top ones are really similar. They all have more or less the same haplotype across this region. So if we phenotype them and they're all susceptible, then actually we could have just phenotyped one of them. So the idea here is that you could go in very specifically to a QTL and now look for new diversity, and then say, well, instead of having to phenotype 400, I maybe only phenotype 5 of these accessions, capturing local diversity. And then, of course, the next step is to use these IBs by haplotypes plus environmental data to see if any of these specific 5 subhaplotypes are enriched in a specific region. And that would work especially well for abiotic stress resistance genes, or QTLs. We tried this again, sorry, jumping a bit here to wheat. We tried the same thing again for a known gene that we were working on in a collaboration with the UK CJR Center. So this is not my background. Janneke Balk suggested these. So these are genes involved in the negative regulation of iron transport. So the idea was that if there's difference in HRZ, we could hopefully increase the amount of iron in the grain. And again, we ran these maps where we used our reference gene and then looked at HRZ 1A and 1B. And here was really interesting to us because on this gene level now, it looked like we have literally no variation. So going down, you have all the different accessions. And again, you can see that the red stripes are basically indicating the introns, but within the exons, there's hardly any variation along these lines. So we wouldn't expect that this is a very good population to find new expression of HRZ1 A or B or functionality. On the other hand, we only concentrated on the allele here. So the next challenge will really be to figure out how big or small to make these things. So we can go from a specific gene allele all the way to a locus. You capture different information depending on what you do. So we're really still playing around with this data. And then the second way that again, I will use wheat as the example, but the same is happening now in barley. I was to say, okay, we can use this really locus-specific things now. What else can we do with these new sets of data that are coming out, and how can we cross-link genomic data sets better? And here, the study from Simon Krattinger at KAUST was really interesting to us because they developed a tool called Missing Link Finder. And what you can do here is that basically he took a pool of k-mers, so the little blue circle here, and asked how much does this pool of k-mers overlap with each accession of the 80,000 samples that were sequenced from the CGRR genebank samples to find out if any of them have a really high overlap. And then this high overlap indicated that they were probably derived or having parts of the Aegilops tauschayi lineage 3. So they used this for a kind of developmental evolutionary study. But we said, well, if you can use that with tauschayi, why don't we use that with any of the crop wild relatives that were sequenced as part of the Open Wild Wheat Consortium? This is maybe also a reminder for the barley consortia that really having these wild relatives is really useful, because you can of course do the same now. So in short summary, what we did here is we took each of the genebank samples, we broke the DATSEC data up into k-mers, and then we asked, well, how many of these k-mers overlap with Timothy? How many of these k-mer overlap with any of the Aegilops species? How many overlap with Dicocoides? And so on and so on. And we got a value of overlap. This value of overlap is known as a Jacquard index. So it just tells you, well, if it's high, there's a lot of overlap. And here you can see the 80,000 CGAR accessions, and you can see this slightly odd linear relationship between the average Jacquard index. So the Jacquard index each sample had across all the wild wheats that we tested versus the specific Jacquard index. In this case, for Aegilops longissima, you can see it's kind of linear. So we think this has a lot to do with sequencing depth. So if some of the samples were sequenced deeper, you just have a higher probability of overlap. So this would indicate to us that none of these samples have a specifically interesting overlap with Aegilops longissima. It just shows you that some samples were sequenced a bit differently. We're still figuring that part out. But what's really interesting then is when we use Polonicum. I think I saw Nikolai in the audience here, so he will hopefully listen closely, because a lot of this was unravelable because of his research. So what was really nice here is that you can now see the Jacquard index. Again, you have this linear thing, and you have a slight population split. So we think that's because we used hexaploid and tetraploid. But then you can see on the top, some of them have really high Polonicum-specific Jacquard indexes. And the interesting thing was when we looked again at the passport data, that a lot of them are Portuguese land races, and there's actually a population of Portuguese land races known to have been derived from Polonicum. They're called Thadas. And there's also TF Francis, which is actually also a Portuguese land races. And then there's something that is actually a Polonicum. It was just missequenced. And at the top, we have this Durum line, but it's from USAID gene bank. So this could actually be just wrong passport data. Again, showing also that this analysis can not only help us to identify land races that already carry some crop wild relative inside them, but also can help us to clean up gene bank data, which is becoming more and more important as we rely on this data to go into gene banks. And then, yeah, another example here again, the average Jacquard against the Aegilops CRC. And we had a few land races from Afghanistan and Iran that scored particularly high. So those ones we would try to do some more in-depth sequencing now to confirm that they actually carry some Aegilops CRC introgressions and could thus be used for bridging. So yeah, the future outlook here of how we could go into gene banks is that we think that be a large effort now onto this fixed system where we could use much better historical climatic data and crop phenology to actually build better predictions on which line would carry resistance for a specific target region or target trait. We should then really try to get away from making hundreds of samples as core set, but maybe actually focus more on getting down to 10 that we can then actually cross and adjust for flowering time, because that's something that's still a really big issue, I think, in the pre-breeding community, that if you score, I don't know, for disease resistant, even for disease resistant, if you score 100 lines from the gene bank, the flowering diversity will just be so great that's often you can't results. It's like compare the results. It's like comparing apples to pears, which is not really fair. And then once you made this cross, you should then screen any derived material. But at the same time, also we think the combination of FIGs and genomic data to kind of do pre-breeding and gene bank selection at the haplotype level rather than on the genome wide, and also to move into the space of reference-free genomics and gene banks, especially because it will also allow you to cross-compare between gene banks, right? So there was a very nice study now from the Swiss gene bank with Beat Keller, where they showed that actually indeed you can compare now data sets from the Swiss gene bank to the German gene bank to any gene bank that has sequenced their barley accessions. And yeah, I think with that, we have the time to come to the second part of this wheel that I showed at the beginning. So we talked a lot about this, how genomics can help to better do this funnel, and where I mostly also used wheat as an example. I'm very sorry about that. I'm still switching to barley. But I would now like to focus a little bit on the second half, where the post-crossing can also really be aided by genomics. And here I'll focus on again, not barley, but on Hordeum bulbosum, which is a great source of novel diversity. And I'm sure some of you have already heard about it, but just to give you some background. So Hordeum bulbosum is the second closest crop wild relative of barley. It originated in the Mediterranean region specifically, and up to Central Asia it can be found. It's a perennial, it's an outcrosser. It can be diploid and tetraploid, and it makes these bulbs, which is of course really cool. And I get really excited whenever I find them. So you can see me here with one in Lebanon in the wild, and I also found one in Morocco, which was especially exciting because there haven't been many sightings of it in Morocco anymore. Also due to habitat destruction, of course. So when I joined ICARA, we ordered all the bulbosum that was available from the genebank, which wasn't that much because it's, of course, as an outcrosser, really hard to produce a seed, and to do all the regeneration is not easy. Oh, sorry, I think my camera gave up. Can you still hear me, Sebastian? Okay, cool. So here you can just see from this handful of about 50 accessions that we were able to order from ICARA, there was great diversity. So for example, we had one accession from Lebanon that had nearly naked seeds. So the seeds were detached really easily. And it had also these very rough own kind of hooks. And then we had a different one that was from Cyprus, which had these very pronounced extra lemma-esque structures. I don't think anymore they're lemmas, but that was really interesting. And of course, this really deep purple seed color. So just showing again that also within one crop wild relative species, you can have a lot of diversity. So only sequencing one maybe doesn't show you everything that's there. And with a PhD student at ICARA, Khadija, we're now trying, of course, to reproduce crosses between bulbosum and vulgare, which are extremely, extremely difficult. They were actually achieved basically systematically only once by someone called Pickering. And we do still try, or we were also in contact with him to help with the protocols, because it's very hard to get the cross. You also then need to undergo high embryo rescue. You need to do back crossing, and yet the chromosomes are not very easy to get introgressions from. However, Pickering managed to actually do these crosses between Golden Promise, Emir, and Murex, so 3 barley varieties, with 11 different bulbosums, some more than others. And these resulted in 146 near introgression lines from bulbosum. So in parallel to our work of trying to recross and get more diversity from bulbosum, we said, hey, let's also use these ones because it's just that much easier. And actually, ICARA had already ordered them and started working on them when I joined. And that was really great because it meant that we already had field data from these near introgression lines. So here in the graph, you can see on the left the biomass of actual field trials. And on the left, basically, is all the lines that have Emir as a recurrent parent. And on the right, you have all the lines that had Morex as a recurrent parent, indicated in blue. And of course, here the interesting thing is to say, well, which of the lines of the introgression are significantly better than their recurrent parent, because that's all we can compare them to. It wouldn't be fair to compare them to elite ICARA breeding material. And one thing we noticed, for example, especially here, is the NILs 105 and 118, which seem to be very high in biomass in certain environments. And this got really interesting because the NILs were not only already produced, but we already knew where introgressions are thanks to the study from Wendler in 2015. So you can see here on the left, the 7 chromosomes of barley, and red indicates where lines had introgressions. So for example, there was one odd introgression line which had the whole of chromosome 7, but then there were also multiple lines which had an introgression on 1H, including our NIL 118 and 105. And then we could now break up the phenotypes we saw by introgression, right? So for example, on the top, you can see the nil 105. It had an introgression from bulbosome 2032. And it had a relatively large introgression, while nil 109 and 118 had introgressions from the same donor line. We don't know which allele, because of course, those donors being tetraploid, they could have multiple alleles of the same region, but we just going to assume it's a similar one. And then you had also nil 103, which again has an introgression in 1H, but from a different donor line. And to us, it was really amazing to see how nil 103 has the same introgression, but behaves in terms of biomass production and flowering time exactly like the wild type, while nil 118 and 109 had quite high biomass and were still behaving like the wild type in flowering time. And then the top one had the high biomass, was also late, indicating that there might be a negative flowering factor, but only in the larger introgressions, something we would like to really keep investigating. And of course, for breeding, we should really use nil 118, right? Because there's the smallest introgression, which still has the biomass effect. But then how would you use this nil 118, right? That's another question we came to. So here again, you can see graphs from Wendler et al. Publication. So here you would see only chromosome 2H, and then yellow again is indicated where according to their GBS data, they assumed the introgression to lie. So this was the basic of the graph I just showed you previously. And here was really nice because, okay, so they used GBS, so they mapped them really precisely, right? But for us at ICADA, that's not really feasible. We can't pay for GBS if we want to cross with this line now, because we want to move this introgression from the EMIR background, of course, into an ICADA elite background. But we're not going to be able to pay for GBS. That was clear. But what we did have was 50K iSelect SNP marker data for the same line. And we noticed that if we now just simply ask, what is the number of SNPs that are identical in this line to the EMIR recurrent parent, we found that across the chromosome, without the introgression area, it's 0.15% difference, right? So it means that basically all the SNPs between the EMIR and the Bill 41 are the same. So they're true of each other. But the moment you move into this region at the end of the chromosome, the number of SNPs that are the same between the Bill and EMIR is only half of the SNPs. So 47% of the SNPs were not EMIR. So this indicated that the SNPs there actually tag pretty much perfectly the introgression mapped as in GBS, and that something like the 50K can be used to trace introgressions, which is of course much cheaper and easier to apply than GBS data. And we tried it for another line that we happen to have 50K iSelect marker for. So again, on 2H, you can now see that across most of the chromosome, the Bill was exactly the same as EMIR. Only 0.06% of the markers were varying, so this could just be sequencing errors, of course. But then in the long arm of the chromosome, you again have the small region where 44% of the markers are not the same, so tagging again the introgression, and also even kind of picking up on that the introgression is smaller. So maybe the 50K iSelect would even be a good option when we make our new crosses, to just test at the beginning if we had any introgressions. Of course, any fine mapping, you would still need the GBS, but there it would also be worth it to spend the money, because it's a resource you're building for many people for many years. So of course, next we said, well, if we now make our crosses, we don't want to run all the 50K. That's still pretty expensive. We would rather just run a few markers. So I went into the region at the distal end of the chromosome and pulled out all the markers that had a variation between the Bill and EMIR, and this variation not being N, because of course there's also a lot of N. So a lot of the markers still don't work on bulbosome, which is expected as it's a really distal relative, but some of them actually amplified. And then, so you can see here now, in the column the markers, but then also the both bills being a C, for example, in the first marker, and 3 elites from the breeding pool having T. So this should even work with a couple of different genotypes and for all the bills. So this is another thing we try to do now, that we don't have markers for each bill, but that we have markers for each introgression. So this marker, for example, at the top should work for bill 41 and 82, not just for one of them. And then I said, well, actually, there's still a lot of markers. I can still save some money here. So why don't we use only the markers that have a G variation, which works much better in CASP application. And then we took those markers and ran them on a cross that actually was already made. So these are F4-derived F5 lines from Miguel's breeding program. And here you can see there was a line from Icada. So this Malu is a pedigree cross to Bill 41. And running those three markers, we found that within this very small population we had, the wild type Icada line had a GAA, while the bulbosum would have the expected AGG. And we found that actually, indeed, 4 lines had exactly the AGG, 2 lines had the Icada genotype, and 2 were still heterogeneous lines, which could now be used to make some more precise introgression backgrounds, or of course, can be now used for selection. So yeah, just to summarize that part, what we really should aim for in pre-breeding with crop wild relatives, out of my insight from it at Icada, is to have actually these kind of precise populations, such as the nil population from Pickering, where you used one elite, and then you try to phase as much of the crop wild relative genome into individual lines with some overlap, and ideally you also have little markers tagging each one, and those markers could be developed quite cheaply. And with that, I think we're okay on time. So I'd really like to thank everyone involved in the work I just presented, especially the breeding team at Icada that looked after everything and helped with everything I did, the INRA collaborators, the physiology team, of course, also the genebank team, as well as the pre-breeding field team, so the guys that actually had to grow everything, and of course also the funders. But if you want to find me, I'm living here now at IPK in this nice little building. So please do come over anytime or reach out for any ideas at the moment. And with that, I'm looking forward to some questions. I don't know what happened to my camera. Oh, I think you're muted, Sebastian.
Yes, thank you, Anna, for your talk. It was very interesting, very nice talk. So now we arrive to the question session, so people can put their question on the chat box or raise their hand to be able to ask their question by themselves. But at the moment, I don't see question yet. And cannot see if there is some hands raised. Maybe I will just ask the time that other question raised some more general question about is your gene bank available for distribution worldwide, or there is some restriction with some regions, or so far anyone that will access to that resource can contact you?
Yeah, so gene banks are definitely there for everyone, right? So talking about both Icada and IPK gene bank, of course, we distribute all around the world. Everything is distributed under SMTA, and some lines are thus also not available. So if you go to the seed store and you see not available, that can be due to seed availability, but mostly it's due to legal restrictions, because we really care about acknowledging that some other person collected these lines, that someone looked after them, and that this should be really manifested. So there's some restrictions on which lines you can order, but in theory they go everywhere, also for free, except that since a few years we introduced handling fees. And we're happy to discuss that if it's for especially developing world research, if we can work on them together. But yeah, there has been a handling fee introduced, also just to make sure people order things they're really going to work on.
Yeah. I asked a following question about this. I saw some years ago a large GBS initiative that had been done on the gene bank of the IPK. So I suspect that most or a large part in barley of this gene bank collection is supported by GPS data, and is all this data publicly available so people can access the seed and also the genetic information to work on it, I guess?
That's a great question, which I would refer you to Niels Stein, who was in charge of that project for the data availability. I think something that we really need to work on is to not only have it available for the handful of experts, but actually to make this data available to everyone, right? And it's the same with the bioclimatic data. I mean, it's nice if you could download that as a matrix or whatever, but actually I think it will be more and more important to build these into the browsers that we use to access GeneBank material. And that's something I'd really like to see a big drive, so that we don't just use this for a nice publication, but really work with it. For example, through Genesis has already pushed this, right? So if through Genesis, it's a platform that allows you to access most gene banks in the world simultaneously. So if you would look for barley there, you would see IPK barley, Icada barley, James Hutton barley, if you are part of it. I'm not sure. Any gene bank is part of it, and they already introduced that you can filter by geographic location, by origin. They're even trying to build AI filters. So I think seeing the same in genomics would be really important, because it's one thing to say, yes, here's all the data, good luck with it, or here's the raw data, or here's a data matrix of SNPs, versus making it accessible to non-bioinformaticians. That would
Okay,
be
thank you. Thank you. Luke raised his hand, so if you want to ask your question, Luke.
Hi, Anna. Nice talk. I was impressed by the bulbosum work. Is it too early to do a fixed approach using the crop wild relatives?
Thanks, Luke. Nice question. I don't think so. I don't think it's too early. Why not, right? Why not prioritize within the crop wild relatives? It's so hard to work with them anyway. You might as well work with the ones that are of interest. And I think in this respect, it's also really great to see that some groups have focused on studying bulbosum directly. So having some insights into which bulbosum might be the best donor for you is also really interesting. And yeah, it has been done in wheat in a study by Eduard Akonov recently, where they did sort of like a fixed genomics approach to identify lines of, I don't know which one anymore. I think of edgelobes to prioritize crossing for heat resilience. So definitely it's possible, and we should think about it, because there's not just one bulbosum, right? There's about 600 that we know of.
Could I ask a follow-on question? Again, a bit more, not necessarily about bulbosum, but using the fixed approach. Is there a way of targeting traits that aren't necessarily correlated with ecogeographical data? I'm thinking of uses like molting quality or
No, I don't think that's a great question. So it works really nice for diseases, because diseases are also linked to actually climatic data, right? They can only occur in certain regions. So I like that jump a lot, and I think that's really fun to work with. Molting quality per se would be a hard ask. Oof, I need to think about it. So one thing is, of course, interesting. So one thing I'd say is that there might be a difference between European land races and other land races, or the ones that were used as donors, but I'm not sure about that part. I just think this might be a use case where it would be way more interesting to go down the genomic route and just say, well, these are the QTLs I know to be involved with molting. Which other diversity is there in the land races for those QTLs, right? Or for those genes? Yeah. Nice question.
No, I mean, I was just wondering if there's any additional data on usage or that could be used to screen.
No, we haven't.
No.
We haven't done that yet. Of course, yeah, you could use cultural. You could add cultural use. It has been very hard to integrate that in gene banks, right? So there is often in the passport some information about what it was used for, or how it was also grown. So, for example, also do the farmers irrigate would be a really important question, because that would really change. Because the data is often not numerical, it's a little bit hard to work with it, but it would be great to see efforts there.
Yeah, cool. Thanks.
Thanks. Nice point.
Thank you. So there is a question in the chat here. I think updating the collection is one of the biggest challenge for GeneBank. If I were to look for seeds from a cultivar released in 2011, I will not find them. I can find landrace and old cultivar, but not the new one. Is there any seeds here to close this gap?
Thanks for the question. So as a GeneBank manager myself now, and also at Icada previously, the gene bank, we were very strict on saying, no, we don't take up breeding material. It doesn't belong in gene banks. Firstly, it could spiral very quickly, right? So once you start taking breeding material, there's a lot. Where do you stop? Secondly, of course, in more close breeding programs, it's mostly a reshuffling of QT of different genetic regions. So how much would you really need to take those ones? And the gene banks are there to save things that we might lose because they're not cultivated anymore, right? That was why Vavilov founded gene banks, because he said, well, industrial revolution in agriculture, but also all the construction side is really destroying natural habitats where people would have used them. So if we don't preserve them now, they might be gone forever. Well, I think breeding material is still really alive, right? It's actively used. Every breeder recycles their program and so on. So I think that's the difference. But the gap I would like to see closed is that breeders also start to sequence and genotype their lines so that they can use them as a basis to go into the gene bank, right? We've seen quite a few I can think of a handful of studies where actually a gene was discovered and a gene was from the gene bank, and then later on it was discovered that the gene is already used by breeders because it's a great resistance gene, so they already picked it up. So to actually find novel diversity and not to re-find already used diversity, we would need to have more genomic data on the breeder side. So maybe incorporating that kind of information would be really fun to see. I hope that answers the question.
I forgot to mention this question was from Jihad, so thank you for your question, Jihad. If there is additional information you want, just put it in the chat or raise your hand. So far, there is no other question in the chat or hand that had been raised, so maybe to give a chance to people to ask another question before we end the seminar. I would have one question to ask you. It's purely by curiosity. Maybe you didn't really look carefully at it, or I just wonder, when you grow the bulbosome introgression line, if you observe any threats that are characteristic or might be partly related to perennial threats that segregate in this introgression line, or you didn't see any attributes of perennial threats?
Yeah, thanks, Sebastian. It would be really nice to know the genetic basis of the bulb formation, right? And yeah, we did check. I did pull out plants. So you only see the bulb formation after flowering. But yeah, I went to the field and checked them by hand, and none of them had any by eye obvious tendencies to this trait. So sadly, yeah, I think there was a publication where they looked at the genes, and they found also flowering genes involved from Israel a couple of years ago. If you're interested, I can send you. Otherwise, I think it may be something we need to map within the bulbosomes.
Yeah. Oh, too bad. Thank you. Okay, so last chance for people to ask a question. Otherwise, maybe we will. I will thank Anna, and we will end this today's seminar.
Thanks, and yeah, just reach out anytime via email or Blue Sky if you have any questions. Thanks for having me.
Thank you, Anna, for your great talk, and thank you everyone to attend for this talk today. Thank you.
Yeah, thanks for coming. Bye.
Bye-bye.